Today's topic is plant tissue culture. Chapter 9. Tissue culture is a method of growing a whole plant from a cell or group of cell or plant organ into a whole plant. That is the plant tissue culture. When a cell is grown into a whole plant in the culture medium under aseptic condition, that is called plant tissue culture. Plant tissue culture is always called in vitro culture. In vitro culture means when any parts of the plant, when a cell or group of cell or a part of the plant organ is grown in the test tube or in any glass vessel, say for example conical glass or petri plates or any glass vessel, if the cell materials are taken and grown into a whole plant, that is called in vitro culture. In the in vitro culture, in the test tube, the culture medium is taken. The culture medium, there are 5 culture medium are commonly used. One is MS medium, second one is white medium, third one is niche medium, fourth one is compost medium, fifth one is knob medium, knob solution medium. These, any one of the culture medium will be taken in the test tube with explant under aseptic condition. That explant can be grown into a new plant, that is the plant tissue culture or in vitro culture. In vitro means when any cell is grown in the test tube, in the lab condition, in the aseptic condition, that is called in vitro culture. The in vitro culture will be followed in the plant tissue culture. There are 3 fundamental principles in plant tissue culture. In plant tissue culture, there are 3 fundamental principles. One is explant should be selected. Second one is explant is isolated. Third one is the explant is culture. Maintenance in the controlled condition. The explant is maintained in the controlled condition. That is physically and chemically condition. Physical the explant is maintained in the controlled condition, that means the physical condition or chemical condition in order to culture. So this is the culturing of explant. So there are 3 principles followed, fundamental principles, considered as the fundamental principles followed in plant tissue culture. One is the explant selection, second one is explant isolation, third one explant culturing. The first method, explant should be collected or selected from any plant organ. Plant organ means root, stem, or leaf, or flowers, or buds, any one of the plant parts, the explant can be selected. What is explant? The part or cell or group of cells or tissues are used in the plant tissue culture method is the explant. The explant refers to any cell which is going to grow, or group of cells, or plant organ which is used in the in vitro culture or tissue culture in order to grow into a new plant. That is the explant. Explant can be selected either from the root or from the stem or from the leaf or any part of the plant. The explant can be selected. Selection should be carefully you have to select. Second one is isolated. Explant should be isolated from a plant pot. They explant a group of cells. Say for example, that should be isolated. Isolation is a different method. Isolation of explant is a different method followed in plant tissue culture by which the explant can be carefully isolated. The third one is maintenance. Maintain in the controlled condition. The explant should be maintained in the controlled condition, either in the physical condition or in the chemical condition. So sterilization is carried out in order to maintain the explant for culturing. Culturing of explant can be carried out in the third process. In the culturing method, 4 culturing media will be taken, as I told you: MS medium, white medium, rich medium, composite medium, and knob solution. Any one of the culturing medium will be taken in the test tube, and with that, the explant is taken and culturing of explant will be started. So that is the third process. Right, to come to the advantages of plant tissue culture. Advantages. Plant tissue culture. First advantage, from a single cell, a number of plants can be generated. One cell gives rise to thousands and thousands of plants in a short period. That is the first one. One cell produces many plants in short period. In tissue culture method, one cell produces a number of plants, thousands and thousands of plants in a short period. That is the first advantage. Second one, used in commercially important plants. Commercially very important plants can be cultured by this method. Third one is used in plant research. For plant research, this method is a very good method to follow. Fourth one is the main application plant tissue culture is the clonal propagation. The fourth method is the main application of plant tissue culture. That means it is the clonal propagation of varieties of plants. Varieties means very important plants, commercially very important, economically very important, medicinally very important. Those very important plants are called elite varieties. Fifth one is endangered plants. Production of endangered plants. Endangered plants are very rare plants. Once upon a time, the endangered plants were abundant on the earth's surface, but during the course of the time, due to the environmental calamities, the endangered plants become extinct. Those extinct plants are called endangered plants. Endangered plants can be regenerated once again by this method. So more number of endangered plants will be produced. The sixth one is virus-free plants. Virus-free plants are produced more in number only by this method. Shoot apical meristem is the one method. Shoot apical meristem. By means of shoot apical meristem culture, we can produce virus-free plants. For example, in the case of dahlia and potato and banana, virus-free plants are produced. Seventh one is germplasm protection. Germplasm preservation. Preservation of germplasm. Germplasm means seeds, pollen grains, and any part of the plant which produce new plant. They are called germplasm. Germplasm preservation is possible by this method. So that is the germplasm. The eighth one is secondary metabolites. More amount of secondary metabolites will be produced by this method. Self-suspension culture method. Self-suspension culture method. By means of this is one of the tissue culture method, self-suspension culture method. By using this method, we can produce more amount of metabolites. What are metabolites? Metabolites are chemical compounds produced during metabolism of the plants. What is metabolism? Metabolism is a process of collection process of, it is a total physiological process going on inside the plant that is collectively called metabolism. Metabolism is a sum of the physiological process, not some total physiological process going on inside the plant cell, inside the plant, that is called metabolism. During metabolism, a number of chemical compounds will be produced. Those chemical compounds are called metabolites. There are two types of metabolites. One is primary metabolites. The second one is secondary metabolites. Primary metabolites are chemical compounds which are necessary for the growth and development of the plants. But secondary metabolites are not necessary for the growth and developments of the plants or microbes or any other organism. They are called secondary metabolites. The secondary metabolites will be commercially very important. The secondary metabolites can be produced only this plant tissue culture method, particularly by cell suspension culture method.